So the chapter number 10, cell cycle and cell division. So okay, growth and reproduction are okay. So the context are we are going to study the cell cycle, M phase, that is significance of mitosis, meiosis, and then significance of meiosis. Okay, so growth and reproduction are characteristics of cells, okay, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. Okay, these newly formed daughter cells can themselves grow and divide, and giving rise to a new cell population, and that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Okay, the cell cycle. Now, cell division is a very important process in all living organisms, right? During the division of a cell, DNA replication and cell growth takes place. Okay, DNA replication and cell growth takes place. All these processes, that is the cell division, DNA replication, cell growth, all these processes, that is the cell division, DNA replication, and cell growth, okay, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, that is the growth of the cell, and eventually divides into two daughter cells is termed as cell cycle. What is cell cycle? The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed as the cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. You can say DNA synthesis is a discontinuous process in the cell cycle. Okay, while the cell grows in terms of cytoplasmic, it is continuous process. The replicated chromosomes DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Now the replicated chromosomes, that is DNA, are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Now, phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture, right? Our cells, like human, it takes every 24 hours, right? This duration of cell cycle can vary from organism to organism, and also from cell type to cell type, even within the same organism, right? So it is cell specific. Like yeast, for example, can progress through the cell cycle in only about 90 minutes. Now the cell cycle is divided into two basic phases, that is interphase and M phase. M phase is the mitosis phase. Now you can see M phase represents a phase when the actual cell division or mitosis, the actual cell division and mitosis occurs, and the interphase represents the phase between two successive M phase. Okay, it is significant to note that in the 24-hour average duration of a cell, of a human cell division proper lasts for only about an hour. Cell division only lasts about an hour. The interphase, that is the preparatory phase, it takes more than 90% of the duration of the cell. The M phase starts with the nuclear division, that is corresponding to the separation of daughter chromosomes, that's a karyokinesis. So the division of nucleus, once they get duplicated, the chromosomes DNA get duplicated, they are getting into the karyokinesis, and usually ends with the division of the cytoplasm, that is cytokinesis. So the interphase, though called resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases, that is G1, that is gap 1, S phase, that is synthesize, and G2 phase, that is gap 2. Now G1 phase corresponds to the interval between mitosis and initiation of DNA replication. Okay, so it is interval between the mitosis and the initiation of the DNA replication. Okay, this is one, the G1 phase, as you can see in the diagram. This one is the S phase, and then there is a mitosis phase. So in between there is a G1 phase, also the G2 phase between the S phase, synthesis phase, and mitosis phase. Okay, as you can see. Okay, then okay, now during G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. Now S or synthesis phase marks the period during which DNA synthesis or replication takes place. DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. The amount of DNA per cell doubles. If the initial amount of DNA is denoted 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell had diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosomes remain the same, that is 2n. The chromosome has the DNA over it, and DNA gets replicated, not the chromosome. Chromosomes are still attached to the centromere. Okay. Now you have to understand that in animal cells during the S phase, DNA replication begins in the nucleus and the central duplicates in the cytoplasm. You see, DNA replicates begins in the nucleus and the central duplicates in the cytoplasm. It happens in the animal cells only. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. During G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Okay, G2 phase, proteins are synthesized. Okay, and cell growth continues. So this is how. Okay, now this is like G1 as G2, then prophase, metaphase, anaphase, telophase, then cytokinesis. This is M phase. Then there are like some cells are uh, uh, exterior to this G1 phase, they exit, that is G0, that is quiescent phase. We are going to study a diagrammatic view of cell cycle indicating formation of two cells from one cell, right? Formation of two cells from one cells. It's shown that one cell forming two cells. Now, how do plants and animals continue to grow all their lives? Do all cells in a plant divide all the time? Do you think all cells continue to divide in all plants and animals? Can you tell the name and the location of tissues having cells that divide all their life in higher plants? Do animals have meristematic tissues? No, animals do not have. Plants have the meristematic tissues, and so they continues throughout their life, and they continues throughout their life. Okay, these are the meristematic tissues present in the plants. And do all cells in a plant divide all the time? No, not all. There are some permanent tissues, there are some simple tissues, there are meristematic tissues. Okay, okay, so only few of them. Do you think all cells continue to divide in all plants and animals? Do you think all cells? No, it shouldn't be continue, because it depends on the requirements. Meristematic, yes, it is. If permanent tissue, then they make the bulk of the body that matures and differentiates. So no, not at all. Okay, so anyways, you have studied mitosis in onion root tip cells. It has 60 chromosomes in each cell. Can you tell how many chromosomes will the cell have at G1 phase, after S phase, and after M phase? So G1 it is 60, S phase it is 60, and even after M phase it is 60. Okay, also, what will be the DNA content of the cells at G1 after S and at G2 if the content after M phase is 2C? If the content is 2C, it means S phase it is 4C, G2 phase it is 4C, and G1 phase it is 2C, because it gets replicated during the S phase. Okay, so now, some cells in the adult animals do not appear to be exhibit division. Example, heart cells, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. Okay, these cells that do not divide further exit G1 phase to to enter an inactive state, that is quiescent phase of the cell cycle. Okay, cells in this stage remain metabolically active. Even in the G0 phase, the cells remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. So it is G0. Okay, as needed to replace the cells of injury. These cells do not divide further and exist, and they exit G1 phase to enter an inactive stage, that is the quiescent stage, that is G0 of the cell cycle. Okay, in animals, mitotic cell division is only seen in the diploid somatic cells. So in animals, the mitotic division seen only in the diploid somatic cells, not haploid. However, there are few exceptions to this where haploid cells, that is honeybees, for example male honeybees. Okay, the haploid cells get mitosis into the haploid male honeybee formation. Against this the plants can show mitotic divisions in both haploid and diploids. Okay, now M phase. Now we are talking about the M phase. Now, so M phase is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Okay, since the number of the chromosomes in the parent and progeny cells is the same, it is also called the equational division. Since the number of chromosomes in the parent and progeny cells is the same, it is called the equational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division, that is karyokinesis. Okay, karyokinesis, that is division of nucleus, is divided into 4 stages. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves following 4 stages. There is prophase, metaphase, anaphase, and telophase. In prophase, which is the first stage of the karyokinesis of mitosis, follows the S and G2 phases of the interphase. Okay, in the S and G2 phase, new DNA molecules are not distinct are formed are not distinct but intertwined. They get intertwined and it's not clearly visible. Okay, it gets complicated. A prophase is marked by the initiation. Now what happens in the prophase? Now when after the G2 phase, when the chromosomes is entangled or intertwined, but prophase is marked by the initiation of condensation of chromosomal material. It gets clear, the chromosomal material becomes untangled. It was tangled before during the process of chromatin condensation. Now this process is chromatin condensation, that it gets in clear. The centromere, which had undergone duplication during the aspect of interphase in the animals, now begins to move towards the opposite poles of the cell. The completion of prophase can thus be marked by the following characteristics events. Now chromosomal material condenses to form compact mitotic chromosomes. Now chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids at the prophase. Remember, at prophase, chromosomes are seen to be like two chromatids attached together at the centromere. The centrosome, which had undergone duplication during interphase, begins to move towards opposite poles of the cell. Each centromere radiates out microtubules called esters. The two esters together with spindle fibers form the mitotic apparatus. Okay, so this is the prophase. Now cell at the end of the prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus and the nuclear envelope. So they do not show the Golgi complex, endoplasmic reticulum, nucleolus and the nuclear envelope. Now talking about the metaphase. So in metaphase, the complete disintegration of the nuclear envelope marks the start of the second phase of the mitosis. Okay, hence the chromosomes are spread through the cytoplasm of the cell. In metaphase, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosomes is mostly easily studied. Okay, at this stage, metaphase, chromosome is made up of two sister chromatids which are held together by the centromere. Small disc-shaped structures at the surface of the centromeres are called kinetochores. These structures serve as the sites of attachment of spindle fibers to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator, with one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole, and a sister chromatid connected by its kinetochore to spindle fibers from the opposite pole. So you can see opposite, the microtubules attach respective towards the poles, and then they and that's what it's written. Like the plane of the alignment of chromosomes at metaphase is referred to as the metaphase plate. The key features of metaphase are spindle fibers attached to kinetochores of chromosomes. The spindle fibers attached to the kinetochores of chromosomes. The chromosomes are moved to spindle equator and get aligned along metaphase plate through the spindle fibers to both the sides. So this is it. Like it was the early prophase, when the nuclear membrane is not completely broken. Now late prophase, you can see the nuclear membrane is broken, and the chromatic condensation occurs. You can see the two chromatids. This is the late prophase, and then there is the transition to metaphase. Like going to the transition, don't get confused, this is not anaphase. Okay, transition to metaphase. Now there is a metaphase place, as you can see in the diagram B, the diagrammatic view of stages in the mitosis. Then after there is an anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as the daughter chromosomes of the future daughter nuclei, begin their migration towards the two opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed. The centromere of each chromosome remains directed towards the pole, and hence at the leading edge, with the arms of the chromosome trailing behind. The centromere does not split. Remember that. Thus, anaphase stage is characterized by the following events: centromere split. No, centromere split and chromatid separates. Okay, the centromere splits and chromatid separate. As you can see in here, anaphase like this, n. It's like 2n, and then it gets like you can see in here. The centromere split and chromatids separate. The chromatids move to opposite poles, right? In telophase, at the beginning of the final stage of the karyokinesis, that is the telophase, the chromosomes that have reached their respective poles, they decondense. First they were condensed, now they decondense. That means they get they again get tangled and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. This is the stage which shows the following key events. Okay, Now, now the chromosomes cluster at opposite spindle poles, and their identity is lost as discrete elements. Okay, so this is the stage. Nuclear envelope develops around the chromosome clusters at each pole, forming two daughter nuclei. Nucleolus, Golgi complex, and endoplasmic reticulum is reformed. So these are the key features of the telophasic phase. You can see anaphase, then there is the telophase, and then there is the interphase. Now there is a interphase. Okay, a diagrammatic view of stages in mitosis. Now cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but cell itself is divided into two daughter cells by the separation of cytoplasm, called cytokinesis, at the end of which cell division gets completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. Okay, furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation starts in the center of the cell and grows outside and grows outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria plastids get distributed between the two daughter cells. At the time of the cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. Okay. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium, that is the liquid endosperm in coconut. Okay, the karyokinesis is not followed by cytokinesis, as a result of which multinucleate conditions arises, leading to the formation of syncytium, that is liquid endosperm in coconut. Okay, now significance of mitosis. Mitosis or the equational division is usually restricted to the diploid cells only. Okay, however, in some lower plants and in some socially insects, haploid cells also divide by mitosis. In some lower plants as well, and honeybees as well. Okay, they are restricted to diploid cells, but still there are some exceptions, right? It is very essential to understand the significance of this division here. No. Okay, about haploid and diploid insects. Mitosis usually results in the production of diploid daughter cells with identical genetic complement. Okay, the growth of multicellular organisms is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. Okay. The cytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of gut, blood cells are being constantly replaced. Mitotic divisions in the meristematic tissues, the apical and lateral cambium, results in a continuous growth of plants throughout their life. So mitotic divisions in the meristematic tissues, the apical and the lateral cambium, results in a continuous growth of plants throughout their life. Okay, now the next part will be the meiosis.